So good morning, everyone. I want to start by inviting you to Grenoble. Because my institute is located on a beautiful campus in this beautiful environment. I have my lab at MB Grenoble, but we're embedded in this beautiful research landscape as well with other structural biology institutes. And there's actually 2 Instruct centers on campus. So if you want to connect cutting-edge science results on your project with some skiing, hiking, you can happily apply to these Instruct calls. Good. But now for the science, I will talk about family of tRNA modification enzymes today. So RNA modifications are very important to fine-tune gene expression. They're important in steady state conditions, but they are especially important adding reversible layers of information to the RNA messages, for example, in infection, under stress conditions, or in response to other environmental cues. Therefore, it's not surprising that all RNAs actually implicated in the process of translation are heavily modified. And this is something we often forget when we talk about translation. So all these little stars indicate modifications, addition to the usual 4-letter alphabet. And tRNAs are actually the most heavily modified species with around 13 modifications on each human tRNA molecule. And today, I think we're only scratching the surface to understand how tRNA modifications and therefore the differentially regulated tRNA pools contribute to the regulation of gene expression. So I'm trying to illustrate here the complexity of these tRNA pools. So in theory, we have 61 codons, for which we only have 45 anticodons that can decode these. But we have in humans over 400 tRNA genes. And these 400 genes that have slightly different sequence, they can be modified by around 170 chemical modifications. And these are all depicted here. And so it's a mind-blowing complexity and combinatorial complexity that is present in tRNA pools. You can imagine these tRNA modifications or RNA modifications at additional letters to the alphabet that change the meaning. And just to bring an everyday example of how a slight modification of a letter can change the meaning. German, schon, schön, French, salé or sal, English, resume or resume. You see, and there's probably many more languages that bring these examples. So there are enzymes that write these modifications on the RNA or on the tRNA. And the tRNA modification enzymes that catalyze these chemical modifications, they face a real problem. So there's some enzymes that want to modify sets of tRNAs or all tRNAs. So how do they bind this whole pool? Because there's slight sequence difference between the tRNAs. And there's other groups of enzymes that really want to target a specific tRNA, so they need to have very precise recognition mechanisms to spot the one that they want to modify. And I'm talking about the latter group today. Naturally, the anticodon loop in the tRNA is one of the most heavily modified regions. You can see all these modifications that are present in the boxes. They can be present in the anticodon loop. And actually, the anticodon loop is the one that pairs at the ribosome with the mRNA. And so these modifications, they can really significantly influence and impact gene expression at the ribosome. And today I'll present on a group of enzymes that do a very specific modification, a very specific set of tRNAs, it's M3C or methyl N3 methyl cytosine. This is present in tRNA serine, threonine, arginine, and the enzymes that write this modification, they transfer a methyl group from a separate molecule called SAM. This carries a methyl group, and this is transferred in the chemical reaction to the cytosine to create this 3-methylcytosine M3C. The problem is that until recently, the exact enzyme mechanism and the catalytic mechanism was unclear. In humans, we have 3 enzymes that write these modifications, metal 6, metal 2, and metal 8, and they have different target sets. So metal 8 targets tRNA serine, metal 2 has 2 targets, threonine and arginine, and metal 8 is a bit the odd one out, it's in the mitochondria. The enzymes are similar, the tRNAs are similar, so we are asking some questions. How do these enzymes actually distinguish different tRNAs? What is the role of their cofactors? You will see in a minute what I mean with it. What is their catalytic mechanism? It's a recently characterized group. And also going a bit towards this design direction, can we actually target some design inhibitors, and can we find inhibitors to them? And yes, I think using a combination of structural biology methods is especially suited to see really this intricate differences, subtle differences, how they bind their substrates, their different substrates. So we started in the lab, we started out with metal 6. Metal 6 is a putative cancer drug target. If metal 6 is overexpressed in patients, it lowers survival. And our collaborators, Rob Schneider at the Helmholtz Center in Munich, they found metal 6 actually as a top hit in a mouse xenograft screen, where they inject or they treated tumor cells with siRNA to deplete, knock down a set of methyltransferases, and then they injected these tumor cells in mice, and they monitored the tumor growth. And so in the siRNA against metal 6, the tumor growth was the lowest. And this implies that if you would treat metal 6 with an inhibitor, that you could potentially find some cancer drug. Together with my collaborator Stephanie Kaiser at the Goethe University Frankfurt, used nucleoside mass spec to determine the actual target of metal 6 of this enzyme. So it could be found that this actually really targets or really writes only the 3-methylcytosine. That this is very specific to tRNAs, and it's not written in other tRNAs. And then they could even boil this further down, that this enzyme only targets serine isoacceptors and not other potential tRNA targets. So it's a very specific enzyme. And this was where we came into play, because now you know the enzyme does this and that reaction, it has a very specific target, but how does it actually recognize the target? So what we actually wanted to do biochemistry and ideally get the structure of this enzyme. So when we just started to express metal 6 recombinantly. We made recombinantly in vitro transcribed tRNA, and we tested their interaction. To our surprise, our results were somewhat contradictory, because this is a binding curve with serine, threonine, and alanine, so one target and 2 non-targets, and you see they bind all in the same range, so no specificity. And also, if you look at the scale, can you really talk of binding? Because there seems to be a very low range interaction. This was puzzling. We tried to get a structure of this enzyme together with our tRNA. We tried a lot crystallography, but we could never get this ligand bound, and this is probably due to this low affinity interaction. But then this cofactor that was mentioned in several publications, SIRS, caught our attention. So if you put SIRS in addition to the metallation reaction, so you mix metal 6 tRNA in this SIRS enzyme, you seem to get enhancement of the activity or reach activity. What is SIRS? It's a serine tRNA synthetase. It's the one that attaches the serine amino acid to tRNAs. So tRNA synthetases are kind of, I would say, the smartest enzymes of the cell, because they speak two languages. They speak nucleotide. They really specifically only recognize one tRNA. And they speak amino acid, because they then recognize the amino acid and attach it to the tRNA. These are really bona fide enzymes, because if they do mistakes, they screw up the whole protein synthesis in the cell. So very smart enzymes. So we expressed also this SIRS enzyme and did an activity assay. And here you see all the controls, metal 6 mixed with tRNA, all the control reaction. And do I have a pointer? Here the most right, so that we get some activity if we add metal 6 and the correct tRNA. But if we, if we add now this SARS enzyme to the reaction, we get a 1,000-fold increase of the activity. So probably this enzyme, the addition of SARS really makes the metal 6 active and makes the reaction going on. Yeah, but how does it work? How does it activate? Of course, we want to have a structure of this. We tried a lot to get this. You know, it's never so easy. Things fall apart on grids. So what we had success with, it was a fusion construct of this probably very transient complex. So we fused the SARS, this cofactor, to the metal 6 and pulled it out from cells. And you see nicely on these curves that we already pull out a nucleotide ligand, which is in that case the tRNA, when we use mild purification conditions. So we pulled out the sample from cells, including the tRNA, and we put it on grids, and we could then finally generate a cryo-EM structure. The structure is a bit larger than what you think, because we are interested in the mechanism of this enzyme, a small bit of the structure. But this cofactor SARS forms an obligatory dimer, and this dimer can bind 1 or 2 tRNA molecules, and this is then what you end up with, a large complex that the majority consists of the serine tRNA synthetase with the 2 tRNAs bound, and the small portion of it is our methyltransferase. Let's take a closer look of our methyltransferase. So this, the methyltransferase is bound to the anticodon arm of the tRNA arm, and it has an RNA binding domain here formed on the top in the dark color that forms a cleft to accommodate the anticodon arm. The variable arm of the serine tRNA, this is very specific for serine tRNAs, is sandwiched between the two enzymes, and here you see again a surface charge representation. This extra domain, actually this was not predicted because it's not a domain as such, but it's 3 motifs that are interlaced into this basic Rossmann fold of the methyltransferase. So together they form this domain that binds the anticodon loop. Yeah, the interface of this complex is really interesting because it seems quite of minimalistic. So part of the interface here is this binding of a base, a flipped out base of the variable arm of the tRNA. Then we have a second contact point, or first protein-protein contact point, reconstituted of this motif that contains two methionines that interact with a loop of the metal 6. So it's a loop in the SARS enzyme that interacts with the metal 6 domain. And we have a second contact, which is here this exposed phenylalanine that binds a eukaryotic conserved loop in the serine tRNA synthetase. So we have only this RNA-mediated contact and these two protein-mediated contacts, which is very minimalistic for a protein complex. And if we mess around via mutations with these contacts, we also abandon complex formation. So although it's so sparse, these contacts are really crucial for complex formation and also for activity. Okay, we got a lot of information from this structure. We know now what is the RNA binding domain. We know how the complex forms and looks, but we still ran into problems because the active site for us did not reveal any obvious catalytic residues. Excuse this mess here on the slide, but it's really hard to bring this 3D active site in a 2D representation. What I want you to appreciate is that there's a lack of obviously negatively charged residues that could somehow be catalytic. So this was one of the problems. The second problem is that this complex, due to its symmetric stoichiometry, is really super hard to access by biochemistry experiments. So if you want to test kinetics, interactions, what do you label? Because it's a transient complex, you always have this Cis dimer, but all the other components, they could be in 1 or 2 copies. So there's really not much room to explore this via conventional experimental methods. So we turned to collaborate with Alessandra Magistrato in Trieste and Jana Opic, a postdoc in her lab, ran molecular dynamic simulations on our complex. So what he did here in this experiment is a dynamic network analysis. In this kind of analysis, the protein or the complex is subdivided, here you only see a portion of it, is subdivided in entities, and then you measure how these communicate with each other to find how within the complex the dynamics, the signal transduction works. And I try to color here the tRNA plus the metal 6 in these communities. And here this is the analysis. So thick lines, they mean there's a lot of communication, and thin lines mean the entities do not communicate so much. And what is really striking is that this anticodon loop, where the modified base is located, is not directly communicating with a cofactor binding. The SAM is the factor that has this metal group. They only communicate via the variable arm of the tRNA and via these N-terminal helixes that I've shown you before, the N-terminal helixes of metal 6. So it's an indirect communication of these two active site components, the metal donor and the metal receiver. So when you have now a closer look on the on this variable arm of the tRNA, the one that is necessary for the communication, and this N-terminal helix in metal 6, they did another analysis of their data, and this was to measure the frequencies of how often these communicate. So this variable arm residues with the residues of the helix. And they compared the simulation of the full complex, including our Cis tRNA synthetase, or one simulation where they just excluded this other cofactor. And what you see here, that once we remove the cofactor, these two bits of the complex, the variable arm and the N-terminal helix of the metal 6, they have much less interaction, they interact much less often. Even in my lab, a PhD student in my lab then assessed this data, followed up on this data, and did extensive experimenting. And this table is a very brief summary of a huge lot of experiments he did. He assessed tRNA binding of metal-6 by fluorescence polarization. He assessed cofactor binding by thermostability assay. And for these mutants here, the tRNA binding by metal-6 alone and the cofactor binding of the SAM or SAH, they were not impacted. But it really impacted complex formation, which he tested by size exclusion chromatography, and he did activity assays, and the activity was also heavily impacted in these mutations. And this really pinpoints that this the SARS, this cofactor, really communicates via the variable arm of the tRNA with these N-terminal helixes to enable activity of the complex. And I want to show you again how really these the receiver, the cytosine and the methyl donor SAM, or here the product of the reaction SAH, are embedded very deeply within the enzyme. And this is our experimental data, cryo-EM experimental data, the map. And in this map, you see beautifully that the methyl group has been already transferred to the base. It's not present anymore. It should be otherwise on the SAM. It would be stick out here. So it's a really beautiful post-catalytic state that we captured. We still lacked information on the catalytic mechanism, since we could not identify via experimental methods any catalytic residue. So this is also something that Alessandra and Jana followed up with, and it might not seem so striking. So this is a movie of the reaction where the methyl group transfer from the, maybe you can replay. No, I don't try. Where the methyl transfer from the SAM to it's replaced to the cytosine, it's not so spectacular. Yeah. In a way, it's just the enzymes provide a geometric scaffold to enable this autocatalysis kind of, and this is quite. Then on the other hand, it's quite spectacular again that the enzymes have such a precision that they exactly position these reactants to enable the reaction. And yeah. Anyway, yeah, good. So I come already to my last slide here. It's again the artistic representation of our complex. Here the tRNAs, they are shown as little delivery trucks as they deliver the amino acid to the ribosome, to the factory, and they carry these number plates, which is like the anticodon. But they are these workers, the tRNA modification enzymes. They are the workers that need to fine-tune these trucks. So these trucks, they are really fine-tuned and specific and modified. And yeah, I've told you today about the metal-6 enzyme, which is such a worker that tunes these delivery trucks so they can fulfill their function optimally. And yeah, if you want to have one take-home message, is that our worker enzyme metal-6 is not very specific on its own. It's not very specific for serine tRNAs. It's just acting a bit lazy. But it has a very smart friend, the serine tRNA synthetase, the bona fide enzyme that recognizes serine tRNAs, and it just takes a smart friend by the hand and takes the help to recognize specifically serine tRNAs. With that, I thank you for your attention. I come to the acknowledgements. I like to thank my group, even Laura from my group, Luciano and Philip, alumni. They contributed to this project. We have collaborators, Stephanie Kaiser from the Goethe-Uni Frankfurt, Robert Schneider from the Helmholtz Munich, and Alessandro Magistrato and Jana Opic from the Trieste. I thank the facilities that enable this work and our funding sources.